New study reveals fetal brain cell origins of autism, depression, and Alzheimer's. Discovery reveals fetal brain cells may hold the key to mental and neurodegenerative diseases. Imagine a tiny cluster of cells in the womb, already carrying the blueprint for conditions like autism, bipolar disorder, Alzheimer's, and Parkinson's. And this is the part most people miss. These genetic instructions are stirring long before we're born, reshaping our future brain health. A groundbreaking collaboration between the Hospital del Mar Research Institute in Spain and Yale University has uncovered compelling evidence that many genes linked to neuropsychiatric and neurodegenerative disorders are active during the very first steps of fetal brain formation. Published in Nature Communications DOI 10.1038 as 41467-61316W. This study challenges the traditional view that such diseases arise mainly later in life. But here's where it gets controversial. We've spent decades focusing on adult brains, testing patients, scanning MRI images, even analyzing postmortem tissue, while the real action might be unfolding in neural stem cells weeks or months before birth. How did they do it? The team assembled a list of nearly 3,000 genes previously linked to mental illnesses like depression, schizophrenia, and anorexia, cortical malformations, and neurodegenerative diseases. Then, using advanced computational models, they simulated how turning these genes on or off would affect the earliest brain progenitor cells. These progenitors are the workhorses that generate neurons and supporting structures throughout development. Studying this embryonic window is notoriously difficult. You can't exactly harvest human fetal brain tissue at will. So, researchers combined data from multiple sources: studies on human brains, experiments in mice, and in vitro cell models grown in the lab. By integrating all these layers of evidence, they created a realistic map of gene activity in each critical cell type and time point. Dr. Gabriel Sanpere, lead author and neurogenomics research group coordinator at Hospital del Mar, explains, traditionally, scientists examine gene variants in adults suffering from mental illness. We flipped the script. We trace these variants back to the very beginning of brain development, in neural stem cells, and saw how alterations there could lay the groundwork for disorders later in life. Dr. Nicola Micali, co-leader from Yale's Pascal Rackett Lab, adds, the moment when neural stem cells are at work is like a crossroads. If certain genes misfire, the entire trajectory of brain growth can shift, influencing everything from cortical layering to cell type ratios. Their simulations revealed specific gene networks that matter most in particular progenitor stages. For example, some genes tied to microcephaly and hydrocephaly were surprisingly active in the earliest stem cell divisions, while others, associated with depression or bipolar disorder, impacted later progenitor differentiation. Even Alzheimer's and Parkinson's linked genes showed early expression patterns, suggesting a continuum between developmental and degenerative conditions. Zoil Mato Blanco, a contributing researcher at Hospital del Mar, highlights the practical outcome. By pinpointing when and where these genes exert their influence, we identify temporal windows and cell types as future therapeutic targets. This opens the door to gene therapies or precision drugs that intervene at the right moment. Understanding these mechanisms is not just academic. Knowing how a single gene glitch can ripple through developing neural circuits could revolutionize personalized medicine, shifting interventions from symptom management in adulthood to prevention at the cellular origin. Still, this raises challenging questions. Could screening for these gene activities in embryos ever be ethical or feasible? And if we can intervene so early, how do we balance potential benefits against the risks of tinkering with the fetal genome? We want to hear from you. Do you agree that our approach to treating mental and neurodegenerative diseases should pivot toward the womb? Or do you think the focus should remain on adult therapies and lifestyle factors? Comment below with your thoughts, disagreements, or wild hypotheses. This debate is just beginning, and your voice matters. Early brain gene activity linked to autism and depression risk. New scientific findings. Imagine if the roots of mental health issues like autism and depression were actually planted long before birth, during the very earliest stages of fetal brain development. That's exactly what a groundbreaking new study suggests, revealing that many genes linked to neuropsychiatric and neurodegenerative diseases are already active in neural stem cells before a baby even takes its first breath. Conducted by researchers at the Hospital Del Mar Research Institute and Yale University, this study was published in Nature Communications and offers fascinating insights into how genetic disruptions during fetal brain development could set the stage for a variety of brain-related disorders later in life. The team examined nearly 3,000 genes associated with conditions such as autism, bipolar disorder, depression, schizophrenia, and neurodegenerative diseases like Alzheimer's and Parkinson. To explore this, they used a combination of human and mouse data, along with in vitro cellular models, to simulate how genetic disruptions might impact the development of the fetal brain. By focusing on null stem cells, those versatile cells that give rise to all types of brain cells, including neurons and glial cells, the researchers aim to understand how gene alterations influence these progenitor cells during different stages of development. Neural stem cells are self-renewing cells found in the developing brain. They are crucial for forming brain structures and play a pivotal role in shaping the brain's architecture. Regulatory networks, on the other hand, are intricate systems of interacting genes, proteins, and molecules that control gene expression and cellular behavior. These networks are essential for understanding how genes function in specific contexts. By modeling these regulatory networks for each relevant cell type, the researchers could assess how activating or silencing disease-linked genes affected the behavior of neural stem cells. This approach helped them pinpoint when and where specific genes are most active, offering a clearer picture of how early genetic alterations might lead to brain structure and function changes later in life. This study included a wide range of disorders in its analysis, from cortical malformations such as microcephaly and hydrocephaly to conditions like anorexia, depression, and schizophrenia. Dr. Nicola McCauley, one of the lead researchers, emphasized, scientists usually study the genes of mental illnesses in adults, but in this work, we discovered that many of these genes already act during the early stages of fetal brain formation, and that their alterations can affect brain development and promote mental disorders later on. Cortical malformations refer to structural abnormalities in the cerebral cortex, the brain's outer layer, which can result in neurological and developmental disorders. The findings of this study suggest that disruptions to gene function in neural stem cells could contribute to a broad range of conditions affecting the cerebral cortex. They also highlight specific developmental windows when these genes are especially influential, which could be critical for identifying periods of heightened vulnerability or therapeutic opportunities. The implications of this research are profound. It not only contributes to our understanding of how early brain development impacts mental and neurological health, but also lays the groundwork for more targeted studies into gene function during neurodevelopment. This could potentially inform future gene-based treatment approaches. But here's where it gets controversial. Could this research shift the focus of mental health interventions from adulthood to prenatal care? And what ethical considerations arise from targeting gene function so early in life? As we continue to unravel the mysteries of brain development, these findings open up exciting possibilities for future research. What do you think? Should we be looking more closely at prenatal factors in mental health? Share your thoughts in the comments below.